So now let's spend a few minutes thinking about these mysteries of what might be called the miraculous and the missing heritability. And these are mysteries because we expect selection to use up, to consume the additive genetic variants by tending to fix the alleles that are favorable under any given selection regime. And as we saw in the last piece, there is selection all the time, pushing populations this way and that. It should be converting the additive genetic variants to changes of the mean trait value. But it doesn't, at least in many cases. This is my favorite example. It's another experiment, but it's really convincing. It's called the Illinois Long-Term Selection Experiment. It began in 1896 and continues to this day. And here's at the bottom a link to the Moose Lab at the University of Illinois, where they are bravely carrying on with no change in the overall trajectory, which is that corn kernel protein content, one figure of merit in the corn growing industry, and oil content, another figure of merit, have both been continuing at the same average rate now for well over a century without any let-up. That's not something theory would have predicted would happen. When they started the experiment at the end of the 19th century, they selected downlines as well, but these were abandoned in the late 20th century because they were just becoming uninteresting. In particular, this reduction in oil content actually strikes me as implausible. I don't see how you could do that and still be alive. You need membranes, after all. And selecting for lower protein content had also kind of been done and wasn't going anywhere in particular. But the uplines continue to rip along, and as I said, and you can check it out if you don't believe me, they're still on average going up, and it's something like their old long-term rate. And the amount of change is humongous in units of standard deviations. They are both up now by well more than 10 standard deviations over their means at the time the experiment started. That's just out of the ballpark, way out of the ballpark. How is that possible? Well, some light began to be thrown on this, perhaps, by the discovery of the missing heritability about a decade ago, when it turned out that modern studies attempting to genomically map quantitative trait loci, QTLs, started discovering that only small fractions of the apparent additive genetic variants, VA, could be accounted for by the QTL that you could find even in the largest genome-wide association studies, GWAS studies. Human height was the first case to be attempted with truly colossal data sets, which were assembled from many medically motivated studies of different medical issues. You can see how people realized suddenly these data had been being gathered unwittingly. Whenever subjects come into a study, their height, weight, and other basic measurements are always taken, and so those data were there, associated with the resulting genotypes, and this provided the material from which it was possible, still is possible, to assemble larger and larger and larger data sets, mapping QTLs more and more sensitively and more and more finely. Here's the data set you saw before from Utah, showing very high heritability of height in male and female Utahns from local families. All right, so there's a lot of heritability that needs to be accounted for. more than 1,000 quantitative trait loci have been identified by these increasingly large meta-studies that have been done over the last 12 years or so, and they're identified by the method we briefly reviewed before. It's linkage disequilibrium with SNP marker loci to a site that appears to tend to nudge height up or down. This is the logic. We saw it before. The marker loci from different parents will be associated with slightly different distributions of the phenotype value if the marker is linked to an actual quantitative trait locus, that is a spot in the genome that is contributing a plus or a minus nudge to the phenotype. But if the marker is not linked to a QTL, then the phenotype distributions conditional on the marker genotypes will all be the same. Right, that's the logic of it. But these 1,000 or so quantitative trait loci for human height together turn out to explain only 10 to 15% of the observed heritability. Where's the rest of it? That was and still is the mystery, even though more and more sophisticated models have been applied to these data sets and purport now to explain something more like 50% of the observed heritability. But even if you buy that extrapolation, well, where's the other half? We're still missing it. So I'm going to approach this, or connect this finding for height with a different question that interests me for many reasons, including that schizophrenia runs in my family. Why is it so common? It afflicts a lot of people. If you think 1 in 200 is a lot, a half of 1%, and it is, it's highly heritable, dramatically lowers fitness. And as a result, the Wikipedia article on schizophrenia, until a few years ago, at least, still contained this amazing statement. There's little doubt about the existence of a fecundity deficit in schizophrenia. Affected individuals have fewer children than the population as a whole. The reduction is of the order of 70% in males and 30% in females. The central genetic paradox of schizophrenia is why, if the disease is associated with a biological disadvantage, and they mean a disadvantage that large, which it is this variation not selected out? To balance such a significant advantage, a substantial and universal advantage must exist. I highlighted that. They didn't in the article, but that's what many people want to believe. There must be some compensating benefit, either to the affected individual or perhaps more plausibly to his or her relatives. And then they conclude, thus far all theories of a putative advantage have been disproved or remain unsubstantiated. And I think that is all still true as far as I'm aware. What I want to question is this idea that in order to compensate for the tragic cost of developing a schizoid phenotype, it wouldn't happen, right? It would have selection would have got rid of it unless there were some kind of compensating advantage that keeps the susceptibility genotypes in the population. Okay, and the heritabilities for schizophrenia are amazing. The best evidence comes from twin studies showing that, for example, the correlation in liability to different schizoid diagnostic categories, including manic and schizoaffective disorders, are much higher for monozygotic twin pairs in the 80+% range for the correlations compared to dizygotic twin pairs where the correlations are under 50%. And here are other ways of showing the same thing, which I'll leave to you to study, but they are quite convincing that genes have a big effect, and the heritabilities must be of the order of 50% anyway. Several conditions considered different diseases by mental health professionals are strongly correlated genetically with each other. So the monozygotic twins tend to be diagnosed with different ones of them much more often than do the dizygotic twins. That's what this table is showing. And this has fueled ongoing debate about the reality of the distinctions. But that's really a side issue for the one we're focusing on, which is just the question whether it really is as surprising as the people who wrote that Wikipedia article thought, that we still have this clearly maladaptive susceptibility to developing schizoid phenotypes, given that selection just seems to be unambiguously against it. So what is the genetic architecture of these susceptibilities? Is it mutations in a few genes with large effects? If so, which genes are they? Or is it polymorphisms at many loci, each making a small contribution? And you can sense that that's where this account of the mystery is going. And I think the people who make the statement, like in the Wikipedia article, are implicitly imagining that it must mostly be caused by mutations in a few genes with relatively large effects. In either case, of course, we'd like to know what kinds of mutations they are. Are they unconditionally deleterious, or do they involve a good news, bad news trade-off? That's the compensating benefit idea. Are they amino acid and nucleotide substitutions or duplications and deletions? That is copy number variation, larger scale genomic changes. Attempts, of course, have been made to use quantitative trait locus mapping approaches to try to find these loci, and they do find significant evidence for modest numbers of genes with relatively major effects on many chromosomes. Here's a somewhat old example of that, and in many cases, these first ones to be found were genes that make protein products that are that satisfy many criteria that make sense. They are associated with schizophrenia. There's linkage. The markers have linkage to a gene locus. They're biologically plausible, and in many cases, this kind of amazes me, they actually show altered expression in schizophrenia. And many play well-established roles in neurotransmission. So here's a cartoon showing the locations of some of these gene products, which are plausible candidates to affect nervous system function. So that all seems to make sense. Although it should be pointed out that something like half of all the genes in our genome and other mammalian genomes do end up being expressed in the brain. So if everything is expressed in the brain, then potentially everything can be thought of, at least in the absence of better, deeper understanding, as potentially contributing to anything that goes wrong in the brain, whether that's true or not. In any case, the same old problem arises here as well. These major effect genes explain just a small fraction of the heritability that we've long known about from twin studies and family studies more generally. So where's the rest of it? Could susceptibility be a highly polygenic quantitative trait, the way height appears to be? This idea was proposed more than half a century ago by Irving Gottesman in several papers, including this one. The idea was it was straight quantitative trait thinking. So they imagined that there's a distribution of susceptibilities. It goes like this. There are people way out in the upper tail are going to be definite schizophrenics, which means they stand a virtual certainty of developing the condition. And then there's a zone where environmental factors, experiences of various kinds, developmental effects, who knows what, may or may not push them over the line. and then the relatives of definite schizophrenics would have a distribution of susceptibilities that would be shifted a little higher than those of the general population. Totally reasonable idea, but again, if that's the case, there's a lot of heritable variation. Where is it hiding, and why is there so much of it, given that selection against schizophrenia appears to be very strong, and it's certainly plausible that it has been strong for a long time. Well, another possibility would be copy number mutations, and a few anecdotes supporting a role for these have been produced. Here's a paper from 12 years ago showing a case of an affected individual who has a deletion that is not in either of his parents. So here the mom and the dad have this inferred copy number from a genome sequencing study, and the offspring has this large zone, which is more than a megabase in size, where actually it's a couple of megabases, at least, where he, I think it's a he, just apparently has one copy instead of two. So a de novo deletion. And it's a sporadic case. Yes, a 2 megabyte deletion, and no one else in their family was affected in this way, so it's plausible that that is a large, if not the sole, cause of his condition, at least a major contributor to it. But there aren't many such cases. It's hard to believe they explain the general pattern, and there are other possibilities. Number one, many genes with alleles of individually very small effect, many genes with alleles of infrequent effect, for example, low penetrance. They only strike a small fraction of the carriers, or complex genetic interactions like epistasis and dominance, and so on. Epigenetics, imprinting, and other environmental forms of pseudo-heritability all sound attractive, but not all of them can easily explain the high levels of heritability that are consistently seen in the twin studies and other forms of direct pedigree, that is, family analysis. And nor can they explain why in other contexts selection experiments like that on the corn do not run out of additive variance, even when mean values have been changed by many standard deviations. But possibility number one, I have long thought, that of very many loci, each with very small individual effects on the trait, in this case susceptibility to schizoid conditions. I believe that that possibility, that hypothesis, can explain all of the observations, at least in principle. There's a paper from 2012 by Matt Rockman on the course website, which is a very entertaining rant on this idea, from which you can learn a lot if you have the time and the interest. I'd urge you to read it. It's a bit dated now, but the underlying arguments don't depend on the current state of knowledge of anything, really. That focuses more on evolution in general than on medicine, but it does apply even if you're interested in these medical cases like schizophrenia. So there is a lot of recent evidence for schizophrenia that it is indeed affected by many alleles of very small effect. SNP heritability studies, which are a new thing. Here's a 2013 paper, which is a big compilation by a giant international consortium that works on psychiatric genomics. It tries to estimate the total variance in liability explained by SNPs, which are used to estimate then the pairwise relationships of cases and controls, even where they don't have known pedigree relationships. Genetic variation is indicated where the case and control pairs are on average more similar across the genome than case control pairs. And that's true, right? They get these substantial estimates of SNP-based heritability for many of these conditions. The alphabet soup, you'll recognize some of the names. ADHD and SCZ means schizophrenia, MDD is manic depressive disorder, and so on. Here's also another study published about the same time that looked at, it was another one of these big synthetic genome-wide association studies looking at type 2 diabetes, myocardial infarction, that is a certain class of heart attacks, and rheumatoid arthritis, celiac disease. I'm sorry, I forget what T2D is. You may recognize it, and SCZ here in red is schizophrenia. And it ends up having on this log scale of number of detected or estimated disease-associated SNPs in this genome-wide association study, it ends up, they're all actually very impressively large numbers of contributing quantitative trait loci, all with means well over 1,000 sites in the genome, but for schizophrenia it's approaching 10,000 sites. It's the mean was 8,332. And this is increasingly looking as though it's the case for very many general phenotypes. These are data on height again from one of the most recent of these mega synthetic studies, Maruli et al. Published in 2017, which I think is also on the course website, and it produces this wonderful plot, which was then new to the height problem and new to me. It's a wonderful picture that makes the point that the minor allele effect size at all of these thousands of loci that I think they identified in this iteration of the study, they plotted the estimated effect size, that is the estimated contribution to either increased height, that's on the going up from 0, or decreased height going down. The units here are in centimeters, okay, as a function of the minor allele frequency, right, the frequency of the more rare allele, and they're color-coded to show the degree of individual significance that they have. And so with respect to height, the picture is very clearly that the alleles with relatively large, that is centimeter scale, individual effects on height tend to be extremely rare. They could only indeed be detected with humongous amounts of data, all these different medical studies combined for this purpose. Their frequencies are so low. And all those with higher minor allele frequencies, let's say of the order of 1% or more, they all have effects of at most a few millimeters. And they're kind of the same, right, from frequencies of 10% up to 50%, which is nobody's minor, right, two alleles of equal frequency. If they're detectable, they have effect sizes on the order of a millimeter in height. And then the ones with big effects are extremely rare. So this says that although these are dramatic, they are not common. And most of the variation in human height is in fact caused by alleles that have really tiny effects on the phenotype. And if that's true, then for the medical conditions from the previous side, we have the outlines of a general model of quantitative trait variation, which says it's highly diffused throughout the genome, all the variation that matters. Okay, so all the alleles with large effects on height are rare, and so they are explaining only a tiny fraction of all the variation. You could take them away, and this human height distribution would look just the same, as would its heritability. The consequence for evolution is that, as we've seen before, if more loci contribute to the additive genetic variation, there is more scope for evolution of the trait itself. And so here's an example I made years ago showing what you could do with 6 loci with alleles at frequencies of half, 0.75 or 0.95, starting with equal frequencies and selecting upward on the trait. You'd get up to, say, 3.1 standard deviation increase of the mean from the starting condition if you selected up by the time you got the allele frequencies up to near fixation. But with 25 loci, starting at equal frequencies, there's a narrower distribution of trait values to begin with. You can move the distribution a lot by just increasing some alleles up to frequencies of like 75%. By the time you get to 95%, you've pushed the mean up by more than 6 standard deviations. That is, it's now completely outside the original range of the trait, sort of like with the corn experiment. And if it's just 100 loci, which we now realize is a very modest number indeed, moving the allele frequencies from around a half up to around 95% moves the mean up almost 13 standard deviations, an unimaginable amount above the original mean. Okay, so then the summary is that large amounts of heritable variation could be caused, plausibly, I think, by very small contributions to the additive variance from many different loci scattered throughout the genome. These small effect alleles could help explain why the additive variance isn't used up by strong continued selection, even in the same direction, which doesn't happen much in nature, but may with respect to some traits, and certainly does in agriculture. That's the miraculous heritability part. Maybe it's not such a miracle, it's just that there's this incredible storehouse of variation sitting, for the most part, unused within gene pools. These small effect alleles could also account for the variation that can't be associated with markers, even in enormous QTL studies, the missing heritability, and much of it is still missing, even for height. Deleterious small effect mutations could drift upward at many loci, or hitchhike with closely linked adaptive mutations, because selection against the disfavored alleles individually at each of these many loci would be weak. And so although Alan doesn't like this argument very much, I think it does potentially provide an explanation for the tragically high levels of susceptibility to schizoid phenotypes that we do seem to have in all human populations. And so, yes, that could, at least part of the explanation for this surprising susceptibility could be in part a consequence of our history of small population size, which allowed just unconditionally deleterious alleles at many of these loci to rise to high enough frequencies that it would take many generations of consistent selection against them at large population size, such as we now have, to actually get them the heck out of there. That would, I guess, be my answer to Alan's objection, that if they're harmful, and the breeder's equation should work, right? It doesn't care about the genetic architecture of the trait, which is true. So, in any case, do we think Wikipedia's assertion is necessarily correct? Is it necessary to balance the disadvantage of schizophrenia with some kind of large and universal advantage. To the contrary, I don't think so. Okay, that concludes our tour of the world of quantitative traits. Hope you've enjoyed it, and good luck finishing the course and moving on with the rest of your lives. Bye.